Hello, everyone. The UT Austin iGEM team is proud to present Octopus, ocean cleanup targeting oil and plastics using synthetic biology. So let's jump right into the problem. Ocean pollution by crude oil and its derivative, plastic, is a pervasive environmental issue. On the plastic side, the rising consumption of plastics in everyday products has led to a global environmental crisis in which plastic accumulates into the biosphere, especially in aquatic environments. This often results in the disruption of ecosystems as animals ingest small plastic pellets. These small plastic pellets are known as nurdles, as shown in the image in the top right. The nurdles are made of different kinds of plastic and are melted down to become everyday products, such as water bottles and grocery bags. These nurdles are especially prone to being spilled, resulting in a huge issue, especially within the Gulf of Mexico, as shown in the image of the map on the right. The key below shows the documentation of nurdles sampled along the Gulf Coast. Since 2018, there's an estimated 300,000 to 1 million plastic pellets per quadrant per square mile. In the status quo, current efforts to address plastic pollution in aquatic environments have used pool nets and leaf blowers, which cause significant erosion and destruction of the environment. While these larger plastic pellets can be targeted through such methods and can be picked up by volunteer efforts, nurdles, being much smaller than the typical plastics, are much more difficult to clean up. And this is made more difficult due to the easy spread by the wind. Thus, there is no efficient means of addressing nodal pollution, and current efforts have focused primarily on preventative care only, with no solution for the existing pollution.
Over a 40-year period, more than 5.7 million tons of oil was spilled into the ocean, causing a lot of damage to the marine ecosystem. Closest to us here in Austin, the Deepwater Horizon oil spill in the Gulf of Mexico in 2010 polluted about 1,100 miles of shoreline and over 57,000 square miles of the Gulf. And most recently, a pipeline leak off the coast of Orange County, California, spilled an estimated 126,000 gallons of crude oil into the surrounding ocean as of October 7th of this year. There are many different methods currently used to clean up oil spills, but the most common are booms, skimmers, and chemical dispersants. Booms and skimmers are more physical methods for collecting or burning away oil spills, but both require certain weather conditions to be effective or even usable. Chemical dispersants, on the other hand, can break up oil slicks into tiny droplets that can be picked up and metabolized by the ocean's natural hydrocarbon-degrading bacteria. These chemical dispersants will work fairly well even in rough conditions, but are harmful to the marine ecosystem due to their highly flammable and toxic nature.
To learn more about the problem and get hands-on experience, we next sought to reach out to experts who have been in the field. First, we reached out to Jace Tunnell, the director of Nodal Patrol. Nodal Patrol is an organization comprised of volunteers who conduct nodal surveys to pick up nodals along the Gulf Coast. From this meeting, we learned about the pervasiveness of nodals as they are found in a variety of ecosystems, including wetlands. This information was particularly insightful because it brought a new target area to focus. Furthermore, Jace Tunnell informed us about the lack of effective solutions for cleanup, with volunteer efforts being the primary area. Next, we decided to get in the field and participate ourselves. Our team went to Galveston and the surrounding areas to collect nurdles and completed a full nurdle survey, which was then entered into the Nodal Patrol database. From this experience, we were able to truly witness the pollution along the coast, and we collected nurdles and subsequently swabbed them to learn more about the bacteria growing on them. Next, we spoke to key environmentalist Diane Wilson, who sued the large plastic plant Formosa Plastics after their negligence and spill in her county. In this meeting, she advised us to focus on plastic pellets that float in water as they are extremely difficult to pick up and evade current efforts, such as the net and leaf blower tactic mentioned earlier. She also supported our project idea as she emphasized that there's ongoing legislation for preventative care, but no actual solution to address the existing pollution. And lastly, on the oil side of our project, we met with Clean Gulf Associates to discuss current oil spill cleanup efforts. In this meeting, we learned of the different chemical dispersants used and their downsides of their use in the ocean.
In order to further understand how nurdles accumulate in the environment, as well as how bacteria interact with them, we made several trips to local beaches and watersheds to clean up nurdle pollution. We collected samples in the Galveston and Point Comfort areas located in the Gulf of Mexico, just a few hours away from our lab. Samples were taken from both marine and freshwater environments. After collecting samples, we swabbed bacteria from the surface of the nurdles and identified bacterial colonies using 16S mRNA sequencing. Several genera of bacteria appeared, including Pseudomonas, Exiguobacterium, Stenotrophomonas, and Pantoea. These bacteria are common in the environment and found in a diverse set of ecosystems. This led us to ask how we could take advantage of such bacteria for our project. Thus, we propose a phage-based solution to hydrocarbon pollution in aquatic environments. Phage allows us to repurpose resources in cells already present in the environment, which eliminates the need for large-scale protein purification. Rather, we take advantage of native bacteria by infecting them with phage, producing our proteins in a flexible and effective fashion. With this concept in mind, we worked to find an appropriate phage for our project. That search led us to P1. P1 is a unique phage and well suited to our project. It is a broad host range phage which can inject DNA into a wide variety of bacteria, including species such as E. coli and genus Pseudomonas. This makes P1 ideal for transferring DNA to a variety of bacteria present in aquatic environments, including species we identified in our nurdle samples previously. P1 is also unique in that its genome exists as a plasmid after injection of DNA. Under normal circumstances, a P1 phage would assemble by packaging this plasmid genome, also known as a phagemid, into its capsid. Occasionally, however, P1 will package other DNA up to 90 kilobases in size from the cell, leading to a modified phage. The rate of packaging of foreign DNA by P1 can be increased significantly by taking P1 self-recognition tags from its genome. One example of this is PAC-A, a sequence we incorporated into our construct shown later. Thus, we decided to use P1 as a vector for our plasmid in order to produce our proteins of interest in a variety of cells in environmental conditions.
To this, we present the octopus solution. Using octophage, our engineered bacteriophage, we plan on delivering plastic-degrading enzymes such as PETase and oil-dispersing enzymes such as RSN2 to bacteria in order to target plastic and oil pollution in the ocean. First, we will create our phage delivery vector, octophage, containing PETase or RSN2. With octophage and using the broad host range of P1, we will be able to deliver the aforementioned plastic-degrading or oil-dispersing genes to a variety of bacteria. After this delivery, these bacteria can now degrade plastic and oil in marine environments.
PETase and metase are the primary focus of our project's plastic degrading aspect. Both enzymes were originally isolated from Itionella sacchayensis, where they form a synergistic complex for depolymerizing PET plastic and breaking down byproducts of that reaction into biodegradable molecules. PETase breaks ester bonds in polyethylene terephthalate, or PET, removing subunits from a PET chain with each reaction. This process results in monomeric mono-2-hydroxyethyl terephthalate, also known as MHET, which can be further degraded by metase. Metase then breaks down MHET into terephthalic acid, or TPA, and ethylene glycol, or EG. Both of these molecules are usable as carbon sources by some bacteria. Note that PETase on its own can actually degrade MHET directly into TPA and EG, but only at a slow rate. PETase and metase are most effective then when used as a two-enzyme complex.
The enzymes that we are using for the oil spill cleanup side of the project all fall under the category of biosurfactants. Surfactants, short for surface active agents, can reduce the surface tension between two usually immiscible substances, like oil and water or water and air, causing them to emulsify or mix. Biosurfactants are surfactants of microbial origin that have a couple other unique characteristics. When in solution, they are typically found in closed conformation, with their polar amino acids on the outside, allowing them to disperse into solution rather than aggregating like most chemical surfactants. However, upon contact with the oil-water interface or water-air interface, they unfold, exposing their nonpolar surfactant-acting insides. When used with oil and water, this causes the dispersion of the oil into small droplets, as seen in this figure. These small droplets of oil can then be degraded and metabolized by environmental microbes that have trouble accessing the oil when it's otherwise clumped together in an oil slick. For our project, we tried a few different proteins, but eventually settled on Renisfumin-2. Renisfumin-2, or RSN2 for short, is one of 6 proteins produced by the Tungera frog during the creation of their foam nests. In previous studies, it displayed remarkably high surfactant abilities, and additionally, we were able to find an identical version of RSN2 with a fluorescent tag called iLove to allow for easier protein detection during our assays and protein production experiments.
We designed our construct using the Golden Gate assembly paradigm. Following that scheme, we chose appropriate DNA parts with the goal of expressing our proteins in a plasmid which can be easily packaged by P1 phage. In this construct, CP25 is a broad host range promoter that constitutively promotes our coding sequences. This plasmid is used for both our plastic degrading and surfactant plasmids, with one plasmid coding metase and PETase, while the other codes RSN2 in the coding sequence region. The PAC-A sequence increases the plasmid's rate of packaging transduction by acting as a P1 recognition tag. Finally, we use the COL-E1 origin of replication. However, future implementations of this project would use a broad host range origin instead. Overall, these parts create a sequence capable of moving from phage to phage while expressing our hydrocarbon remediating proteins. Our selection of parts were then assembled using standard Golden Gate assembly procedures. Cells containing this assembly can now be infected with P1, leading to packaging of the assembly into P1's capsid. These phages can then inject this plasmid into cells, introducing sequences for our enzymes of interest and leading to their expression by a variety of potential host cells without any need for human intervention. Thus, this system can be used to spread this genetic information into a population of cells in an environment, combating oil and plastic pollution.
After we finished formulating our plan for the phage and delivery system, we had to design assays that we could use to test the systems. For all of our assays, we needed a positive control, in this case, purified protein, to help calibrate the assays and put the results in context. We overexpressed our proteins using IPTG-inducible expression from PET plasmids and purified them using 6 histidine tags. However, the only protein that we were able to successfully induce and purify was I-love-RSN2. This histag purification is shown to the right. As you can see, there are very strong bands present in all 3 elution steps, slightly larger than 25 kilodaltons, consistent with the expected protein size of 26.7 kilodaltons. This suggests that high concentrations of I-love-RSN2 were successfully purified and was confirmed by the highly fluorescent nature and bright yellow color of the eluted samples. For the remaining proteins, we have repeated the induction step with several different strains of E. coli. We started out with BL21DE3, which only worked for I-love-RSN2. The same was true for Lysy-IQ and C41, which were supposed to account for the possible toxicity of our plastic degrading and oil dispersing proteins, and Sheffield E. coli, which provides a reducing environment for proteins that are to be excreted. We also attempted this procedure at a wide range of temperatures, from 16 to 37 degrees Celsius, with no positive results for any of the other proteins.
First, we created our negative controls. We needed a baseline to demonstrate that our system had actually caused plastic degradation. To do so, we used scanning electron microscopy, or SEM, to show the original texture of the surface of our PET plastic. The image on the left shows the PET pellet with no exposure to anything at all. The image in the middle shows the SEM of plastic that was incubated with LB media for 36 hours. On the right, we have the SEM of purified wild-type petase as a positive control. As we can see, there is a marked change in texture, which signifies degradation. Next, we further assessed our positive controls. In general, we had 2 positive controls: purified wild-type petase and a culture of Eidionella sacchensis. With these controls, we conducted HPLC. The purified wild-type petase was graciously obtained from the Alper lab. HPLC, or high-performance liquid chromatography, is an analytical technique used to identify and quantify components in a mixture. Thus, we used HPLC to quantify the release of PET monomers, TPA and EG, as a measurement of degradation. Therefore, greater concentrations of PET monomers would indicate greater degradation. We used 4 experimental conditions: wild-type petase at 30 degrees Celsius, at 16 degrees Celsius, and Eidionella sacchensis at 16 degrees Celsius and at 30 degrees Celsius. These temperatures were chosen to account for the maximum efficiency of petase at 30 degrees and the ocean environmental temperatures, which is at 16 degrees Celsius. We observed strong degradation from purified wild-type petase. However, there was little degradation for the Eidionella sacchensis culture. This result was expected, however, since Eidionella sacchensis uses PET monomers as a carbon source, and therefore very few PET monomers should be detected in the liquid sample. Thus, this does not indicate that no degradation has occurred, but rather it suggests that an additional means of assessing degradation, such as by weight change, is required.
For our experimental sample, a PET film was placed in cell culture with petase-producing cells containing our engineered plasmid for 7 days. The film was then removed from cell culture, thoroughly rinsed, then dried. We then used SEM to image the sample surface. Petase's activity on PET caused it to develop a rough, patchy surface with craters 2 to 5 micrometers in diameter. The top figure is the previously displayed negative control, and the bottom figure is PET degraded by our plasmid. Thus, we have verified our plasmid's successful function in expressing petase and metase.
We used 2 different assays to test the surfactant abilities of our samples. The first, the oil spreading assay, directly tests the oil dispersing abilities of a sample. The sample is added to the center of the plate, containing a thin layer of crude oil covering a much larger volume of water. After a few seconds, the oil disperses away from it, creating an oil-free region in the middle of the plate. The area of this spread directly correlates to the concentration of surfactant in the sample and its surfactant abilities. BSA, a non-surfactant protein, served as our negative control and did not create any spread, as seen in the top left. Triton X100, a chemical surfactant, served as our positive control, creating the large spread seen at the top right of the image. When we tested iLoveRSN2, there was a small spread. We hypothesized that this reduced dispersion compared to the previous data regarding RSN2 is due to the iLove tag. Surfactant proteins conformations are very important due to the unfolding mechanism they use when reducing the surface tension of an interface. So the addition of the iLove tag to RSN2 may interfere with its ability to disperse oil. A more sensitive test for surfactant abilities is the foam stability or bubbling assay. This assay takes advantage of the surfactant ability to decrease the surface tension at an air-water interface. It involves bubbling large amounts of air through the sample and then determining the stability of the foam created by checking how long it lasts. Because the air-water interface is more miscible than the oil-water interface, this assay is more sensitive to surfactant abilities than the oil spreading assay. We used the same samples as the oil spreading assay, but got significantly better results. In this figure, we show BSA, Triton X100, and iLoveRSN2 at the start and 20 minutes later. BSA did not create a stable foam, forming large bubbles that popped quickly. Triton X100, on the other hand, created a stable foam with small bubbles that popped at a steady but slow rate until the last layer, which remained stable for over an hour. When we tested iLoveRSN2, we observed similar foam creation and stability to Triton X100. Although difficult to make out, a single layer of foam remained after 20 minutes for both Triton X100 and iLoveRSN2. This layer, although not pictured past 20 minutes, did remain stable for over an hour. The similarity between Triton X100 and iLoveRSN2 indicates that the iLoveRSN2 can function effectively as a surfactant protein.
To better understand the feasibility of our proposed solution for oil and plastic pollution, we modeled phage infection cycles in a sample of seawater in environmental conditions directly after introduction of a single phage to the environment. In the graph shown, the purple line represents population of phage. The blue line represents population of cells susceptible to infection by our phage, and the red line represents cells currently infected by our phage. This model shows that phage reproduce rapidly over a span of about 100 hours before reaching an equilibrium population close to zero. Production of our proteins of interest then is most rapid during that spike in phage reproduction, after which levels of those proteins reduce due to denaturation by environmental factors. Thus, our solution can act upon a sample in just 100 hours, at which point activity occurs at a decreasing rate. This shows that our proposed solution can respond rapidly to issues in oil and plastic pollution.
Overall, our team has successfully completed numerous experiments. First, we purified iLoveRSN2 in significant quantities. We also proved that our purified iLoveRSN2 can function as a surfactant. Furthermore, we tested our phage construct and proved its success with SEM. We also assayed numerous positive controls using HPLC and SEM. For oil assays, we successfully conducted the oil spreading and bubbling assays. Finally, we also modeled our proposed solution with our phagemid. We are currently still working on implementing our phagemid into our phage delivery system. For gold medal requirements, we successfully completed integrated human practices through our meetings with experts in the field. We also successfully modeled our implementation of our proposed phagemid system. And lastly, through Instagram posts, we completed the education and communication gold medal requirement. Thank you so much for listening. We'd like to give a special thanks to the Alper Lab for providing purified enzyme and HPLC help, the Harshi Lab for providing fresh P1 phage stock, and the Barrick Lab for providing mentoring and lab space and resources, all of which our project would not be possible without them. And we'd like to extend a special thanks to Dr. Jeffrey Barrick, Dr. Dennis Mishler, Cameron Roots, Hannah Cole, Hong Yun Liu, Victor Lee, Kate Elston, and Sarah Bialik. Thank you so much.